the next lecture. So in the morning you have already seen a great example of actually how you can use modeling to understand biological systems or even just systems that are composed of very simple components that are basically forming the basis of life, for example, lipids. Or if you could just consider a molecule that would be basically in water, the most important fluid on Earth, that forms also the basis of life. What I would like to take you now again is to actually introduce you another model membrane system that can be useful. It has advantages and disadvantages as every model system. And we will basically go a little bit into the world of nanodiscs. So if you consider, we have talked quite a lot about liposomes. You now know that liposomes can be in different flavors: small, large, giant, unilaminar, multilaminar, multivesicular. And that is all useful, in particular when you are interested in the type of compartment separation. If you want to have studies where you want to have two different environments. For particular studies, it might be interested to have an open system. In the ideal case, you would say, would it not be beautiful to have just a piece of membrane that you can study? And the reason for that is that if you keep this soluble in a solution, you can use all techniques that are already existing for components that are free in water, for instance, cytosolic proteins that are unbound. We have a lot of techniques to study these proteins. And now if we consider that we would basically be able to do the same with membrane proteins, wouldn't that be fantastic? The problem was with the membrane proteins, they like two environments. They like the water phase, the aqueous phase, but they also like a hydrophobic environment that is given by the membrane. So what would we need to do? We would need to basically keep this type of membrane, a membrane sheet. What is the problem to keep such a sheet in a solution? The problem is that part of the sheet is very hydrophobic. You have basically here lipids that are facing with their head groups the polar environment. You have lipids on the other side, because it's a bilayer, that are facing the aqueous phase. But you have here in the middle, you have this hydrophobic core that doesn't want to be in contact with water. So normally these particles, if you are having them only on lipids, they would never stay in this type of structure in a solution. They would either start to aggregate or rather form a different structure in terms of, for instance, vesicles or types of inverted vesicles, depending on the lipid type you have selected. So people came to thought about how can we basically stabilize just a sheet of membrane? How can this be possible? And there's an example in our blood where lipids basically need to be transported also from one part of the body to the other. If you think about your nutrition, no? You eat basically material that is broken down in your intestine, is taken up, and from these intestinal cells it needs to be distributed to the liver cells, to the heart cells, wherever. And among this material are also lipids, but they are not water soluble. So nature needed to develop basically a trick. And the trick is that nature, and you see it here also illustrated, is using the help of proteins to actually cover the hydrophobic part. And this type of protein is also used in our body, comes basically as a part of lipoproteins that are actually swimming. Okay. Super, thanks a lot. So what we are basically taking is we take the lipid sheet. Problem is this hydrophobic side. And we take a protein. It looks like in the free unfolded like that, but it can wrap around. What type of property would this protein then need to wrap around basically such a particle? It would need a phase that would be basically like to be hydrophobic, and it needs a phase that would like to be hydrophilic. And that's exactly the property of these proteins. Of course, this model is a little bit not perfect yet. If we want to make it perfect, Tanmay, you can stay there. You only need to hold it. We make now, this protein falls into an alpha helix. You need to keep it, yes? It looks like that. And depending which amino acids you have placed, you can have a polar, a water, basically, amino acid that likes water. You can have a hydrophobic amino acid, an amino acid that likes to interact with hydrophobic thing. And if you do this now like that, this side, you would have basically on that step an amino acid hydrophilic. Next amino acid follows hydrophobic. You get a two-phase protein. This one would like to be around an hydrophobic phase. And on the other side, it would face the water. So you make your membrane sheet water soluble. And these proteins are called belt proteins. Okay? They are belt proteins, and they exist already in nature because nature needed to solve such a problem, how to transport lipids in an aqueous solution in form of these belt proteins. They are also called lipoproteins. And we basically use this type of material that already exists in the nature, but we modify it to the minimum that they only have this property of hydrophobe, hydrophile, to cover a membrane. In reality, these proteins are a little bit more complex. They contain additional domains that are important for recognizing, for instance, receptors. We are not interested in that. We only take the core. So we take a type of shortened protein that exists in nature and has this fantastic property to basically bind hydrophobic particles, lipids, and to be hydrophilic to the aqueous environment. And now you have a particle that is water soluble. Okay? And why would this be interesting? Now that could be already interesting just to study membranes by itself, but we need to then have a look. So this would be basically here our little membrane piece that is stabilized by these belt proteins. So a site of membrane scaffold protein, also called MSP proteins. A membrane scaffold protein having this property of hydrophilic areas, hydrophobic areas. Now the size of such a particle is about 10 nanometers. So there's not so many lipids in. You can calculate. Remember, we basically set the head group, it's around 0.5 nanometer. You are all very good in mathematics, so 10 nanometer, you can calculate the area. You can calculate now with the size of a lipid, how many lipids would fit in, around 70 lipids. So it's not large in terms of a membrane, but it's good enough to mimic a membrane for many purposes. It's a small piece of membrane that you have created. Okay? And of course, what you can do is basically you can also decide to insert membrane proteins in this. That goes on the cost of lipids. If the protein is very large, there's hardly any lipid left here. There will be maybe one ring of lipids. If you take a more slim person, unfortunately this is my belt, so but let's say would be here. Here you get a problem. No? If I would basically want to put more lipids in, there would be an area that is not shielded. That cannot happen. So you get thermodynamically, of course there will be dynamics, so like that. But here's still some space, you see? So here I could still have some more lipids. But there's a limit. Huh? Okay, so that allows you to add proteins in. But because of the size, there's a limit. If you have very large protein complexes, maybe of integral membrane proteins, that would not fit anymore in this. But many, many proteins that we are interested in, this system already, it's very nice. So the advantage is you can basically have access to both sides. Why could this be interesting? Remember, for instance, on these receptors yesterday, we had talked about proteins that would basically on one side bind a ligand and on the other side bind a signaling molecule. And that you can now do. Because you can just add the signaling molecule and the interacting protein in the same tube, and both components will have access to the membrane protein. The hormone can bind, and then you can study whether the interacting protein also binds or not, whether the receptor becomes active or not. You can already think about, no? If you now add an inhibitor, you can test, does this protein still bind, or does my ligand still bind or not? So maybe you have actually in this way already the ability to screen for inhibitors. So there's quite a number of advantages of using this system. But just as a warning, it's few lipids. It's not thousands of lipids that are around. There is a size limit in the proteins that you can add. And now comes another problem. We talked about that some of the proteins that we are studying are machines that are actually motors. That means they make movements. Remember, now we talked about movements like that. And if I take this here, you might, so I take a very short one. I cannot move anymore. So then the system is not good to study my dynamics as a protein because I have problems to move. While I'm like that, I can move. Okay? So the system is nice, but it also restricts eventually particular dynamics of the protein or makes them more difficult. So the energetics might change in such a system. You see, typically it requires 2 of these belt proteins to really cover. So you will take the one belt and the other belt just from the size that you really cover the hydrophobic part and that you would be soluble. Now you can make tricks because if you have it like that, the system is a bit flexible, right? It can be useful if you want to have a protein that really needs to move, but it also makes these membranes unstable in a way. So people decided also to make these belt proteins like that. So the 2 ends are linked. Now it's quite stable. With the disadvantage, it might also be restricting again movements and the fusion of lipids and so on. So you need to decide do you need it very stable or do you need it more flexible? And you need to decide, how large you want to have it. Okay, so what you can basically do is, to get these nanodiscs, the prerequisite is you need these belt proteins. Typically you can now buy them, but you can also ask the biochemist to basically purify those, to produce them and purify them. You need the lipids. Lipids you can buy, or you can extract them from your material that you are interested in. Remember, we talked about how to isolate lipids, for instance, by an organic solvent, and then you have a lipid extract. You can also decide if you want to start with simpler mixtures, first these complex lipid mixtures to separate the individual lipids into different classes and start maybe with one type of lipid. Yeah. And the membrane protein, that depends on which protein you are interested in, with which lab you are maybe collaborating. They will typically provide. Or you want to dive into biochemistry, then you learn how to make your own protein by basically using organisms to express it, and then you'll be trained to isolate it from the membranes. There were, remember, 2 important steps. You need first to solubilize this protein then out, and then you need to add it back to form a nanodisc. And that's something we need to discuss, how to do this. Okay, so now what we can do with this system, it's basically there's different disc sites available. It's not dramatic, but still you can make small discs, 9 nanometer, up to 13, 14 nanometer, and that depends on the length of your belt protein. You cannot make it very, very long, because what the problem is, then it becomes very unstable, the system. But you can basically have a long one or a shorter one, and you can decide this for the protein that you want to study or for the membrane piece that you want to study, which type of belt protein you like to take,
to basically assemble them. And what typically now happens is, if you work basically with one defined build protein, so you have hundreds of those components in the same length, you will get hundreds of these particles all the same size. So we call this as a monodisperse preparation. It's not like with the vesicles. Remember when we talked about vesicles, and maybe you have also seen it in the practical part with Rasheni, when you prepared giant vesicles, they all had different sizes. Some were 10 micrometer, one were 2 micrometer. That same happens also on the LOV level. So we will make this afternoon some LOVs, and in average they have 200 nanometer. But if you look really individual, you have vesicles of 100 nanometer, 200 nanometer, 300 nanometer, and we call this heterogeneity. It's polydisperse. Now with these nanodiscs, you have these monodisperse, you have a one defined size. That's very helpful often for many, many techniques that you are using to have a defined size of your system. And that allows you then, for instance, if there are changes in the size, to conclude whether you have a membrane protein, a membrane protein complex formed, or whatever you are interested in. Okay. So techniques that you can use now is you work basically with techniques that are normally designed for soluble proteins or soluble matter, because now you turned your membrane protein into something soluble. So you can put it on an atomic force microscopy. You have seen this also with Tripta's basically talk. That's a technique where you basically use a tip and you actually go carefully over the surface. And once you hit here, for instance, a particle, you need to go basically with the tip a bit up and then again down, and that can all be basically detected. So if you would have nanodiscs here lying around, and you would go with the tip, this tip would basically give you the size of these particles. You can measure on the change of the output of this caterpillar what would be the size. Electron microscopy, that's basically a technique like the light microscopy, but just using electrons, which was also introduced in this course. You would need electron microscopy because what was the reason? To visualize these nanodiscs, why would you not be able to just look at them under the microscope? There was one particular important aspect that you need to consider. It has something to do with the resolution. No? The resolution depends on the wavelengths of the light, and light microscopy, the limit is 200 nanometer. You cannot resolve particles below this, but electron microscopy can this. So that's why you would need, for 10 nanometer particles, you would need to use electrons doing. So you can do small angle X-ray scattering, a technique that we go a little bit more into detail tomorrow. It's a scattering technique that gives you basically information about the structure of the particle. You can use nuclear magnetic resonance spectroscopy that can give you some insight also of the structure of the particle, of the protein, or of the whole complex that basically you can study in this technique. And you can basically use these type of nanodiscs also to put them on a surface. And you can then measure if your particles interact with other particles, with other molecules, how stable this interaction is. Because these techniques, the surface plasmon resonance and quartz crystal microbalance technique, they are very, very sensitive to any change in the surface. So basically, if I would immobilize Tanmay as a nanodisc, that I can first see because the surface has changed. Now he sits here on a surface which was before empty. If he gets now basically a protein to bind on the membrane, on this membrane piece, or you have inserted a membrane protein and the membrane protein binds to the membrane protein, now he has changed his weight. He sits on a surface, but he is now heavier. And these techniques can detect this. It has something to do, I think, those that are physicists can even explain it very easily, because plasmon resonance is basically a technique where you use light to induce changes in the electrons on the surface. The electron starts to oscillate. Yeah? And this oscillation is very sensitive to any change, whether it has something bound to the surface or not. You can, and if you consider oscillators, you know that oscillators are sensitive also, no, to the weight, so that you can detect. Quartz crystal microbalance is the same. It's a quartz that you set in basically vibration. Sometimes this is also used for some clocks, no? These quartz clocks. So you set it, this vibration is very constant, but if you add something on this crystal, this basically changes will change. The amplitude and frequency will change, and that you can detect. So any change on the surface can be detected, and we as biologists don't care so necessarily how this exactly is detected. We are only interested, of course, how the changes are then depending whether there's a nanodisc, whether there's a nanodisc with a protein, or whether there's a nanodisc with a protein, and you added a protein, you added an inhibitor or whatever. Okay? Normally, if you would have a soluble protein, you would not need this. The soluble protein, you can make a surface that it's attached. It's free swimming, it can attach on these sensitive surfaces, and then you can already measure this. Membrane proteins, because of these hydrophilic, hydrophobic properties, you need to provide them with this type of little membrane environment. Okay, so how to make them? So the good news is, you don't need to worry. Why? You take the property of nature. It's a self-assembly process. We talked about self-assembly processes already. There was basically the secret when you take lipids in detergent and you take protein in detergent and you mix them. And now you remove the detergent. What would happen? They would join. And what is the reason that they join? There must be some physics behind, no? Otherwise, I mean, normally we would like to have a very random system. We don't want to have order. Any order costs energy. But we just mix them, remove the detergent, we don't put any energy there in, and still they basically start to form order. No? And which entropy is actually there the most important in the whole? I mean, it's the overall entropy, but on which molecules are the entropies most interesting to look at? For the water molecules. Because the water molecules, they want to be basically free. They want to move. But if you have basically a hydrophobic part exposed to water, then you start to get water order. You know? And so what will happen is, and we also talked in the course, the lipids want to prevent the interaction with water on their hydrophobic parts and only want to expose the hydrophilic parts to the water. And that's why when you give lipids with a detergent and water and you remove the detergent, they are not longer soluble, they need to form a structure. And the structures that can happen depend again on the shape of the lipid. And if you do this with the protein, with a membrane protein, what would be there the problem? It's the same problem because the membrane protein, hydrophobic part, hydrophobic part, this part doesn't want to interact with water molecules. It would structure water. This is basically costly. So we basically get a self-assembly. So now when you basically add this scaffold protein, this belt protein, it would have the same property, right? It is basically hydrophilic, hydrophobic. So if you just take this belt protein in water, what would happen? They would aggregate. With their hydrophobic parts, they would come together, and with their hydrophilic parts, they would show to, huh? But now, of course, we not only give the belt proteins into the solution, we give the belt proteins plus maybe our membrane protein of interest plus the lipids. And what is the role of the detergent at that stage? So we have detergent here, we mix all three components. What was the role of the detergent here at that stage? To keep everything in solution. Because what would the detergent molecules do? They would basically go with their hydrophobic parts to the hydrophobic parts of the belt protein, to the hydrophobic part of the lipids, to the hydrophobic parts of the protein. So and expose the hydrophilic part to the water, so they make these components soluble, so we are in the solubilized state. Everything, detergents, is together with the scaffold protein, detergent is together with lipids, detergent covers our membrane proteins, and since I have so much detergent, it's an excess of detergents, I even have free detergent molecules, and you can consider this like a soap. Now when you remove the detergent, the components have a problem. You have basically the hydrophobic parts free of the membrane protein, the hydrophobic parts free of the membrane protein, hydrophobic parts free of the lipid, hydrophobic parts of the bed protein also free. And they will come together to minimize this hydrophobic exposure. They will aggregate, but they do it in a very smart way, right? Because they aggregate to something like that. That sounds almost like a miracle, right? And I tell you, this also only happens when you have the right mixture. When you have the right mixture of the number of bed proteins versus the number of membrane proteins versus the number of lipids. And you typically see already that when we basically mix this and we remove these biobeads, you get this self-assembly, but as a result you get discs with protein, without protein and with protein. So at that stage it will be heterogeneous. Maybe also we talked some bed proteins might aggregate, right? Some lipids might also aggregate. So it's not that basically just mixing it and voila. It depends whether you have the right amount of each component, and still it will be at that state heterogeneous what is formed. That's important. So, and now to basically remove everything what is not integrated, like free bed proteins, free lipids, whatever that have aggregated, we use a size exclusion. Are you familiar what it means, size exclusion? It's basically a technique where you use a type of porous material. And this porous material has channels, like this. Also like this, right? So these are large channels here, right? I can easily go through this. But there are also channels that, and actually here you see the channel is so small, I even don't fit. I cannot enter. So everything what is large can easily go to these large pores. Everything what's small can actually not go here, but can also go here. And it will start to go here and there. So everything what is small in size in terms of these channels will stay long in the column, long time, while you are flowing basically the buffer through. Because it has all these possibilities to go to all these channels. These nanodiscs, 10 nanometer, they are large. They are large compared only to the free protein, or only compared to one lipid or two lipids with detergent. So they will basically go very fast here through. They can spend any time, they don't have the choice. There's only one choice to go. Why the small material has a choice where to go, and that's why it takes very long. They will explore all possibilities. So that we call size exclusion. And based on that, we can basically separate these 10 nanometer particles away of whatever would be free detergent, free lipids still, or free proteins. And you have to do this because this self-assembly, it's random. You not only get basically nanodisc forms, and you not only get nanodisc formed with protein, and in fact here you get a mixture. Okay. So in that, for instance, here you see even there is a slight difference in the size when you have a nanodisc that would be empty. That's an elution profile. That's basically you let the column flow through, and the longer you wait, up a certain moment, they actually, free nanodiscs, they come later than those that are containing a protein, because they are larger. Those ones contain on top a protein. So if this would be the nanodisc and I would be the protein, it would be a large particle. If you only have the nanodisc, so I'm disappearing, then it's a smaller particle, and the smaller particle needs more time to pass. So you can basically isolate these particles. You can isolate, but there's a partially overlap with this. So that's not a perfect isolation, but at least a little bit. And then you can look here in an electron microscope. 100 nanometer, here the scale. And then you see particle. Of course, when you look at this, you know, as a biologist, so only dirty stuff. I only see dirty things. Is there any, where's my nanodisc, right? But when you then a little bit zoom in, you might start to appreciate to see here something, right? But it doesn't really look like a nanodisc, right? It's a, and in fact, if you look closer, it's 2 nanodiscs together. That was actually a surprise for us because it starts already to show that maybe even in our system, when you make these nanodiscs, they might come together. And that might be due to the protein, and that can be on one hand interesting, but it depends on your questions that you want to answer. It can also be disturbing. So it can be that even in the system where you have now created these free nanodiscs, where I told you wonderful, these little particles, they swim freely in the solution, that this is not completely true, because if these particles contain your protein of interest, they might start even to interact and to attach each other so that you get 2, 3. So that's almost here, you know, we think it's actually something like at minimum 2, where the protein sticks out a little bit. We are not absolutely sure, but we found many of these, when you look, many of these particles not as single particles, but rather as doublets. Okay, so now you might say, okay, it's very heterogeneous. I work basically with a sample in the end that contains basically some of the nanodiscs contain my protein, some not. In particular, if you are interested, like for the microscopy, for structure, that's not nice. You would like to have basically only the preparation that contains your protein, or you might only want to have particles that are free of your protein. So you need somehow to separate. And that's difficult, but one point would be, again, something like affinity purification, a process that we already discussed for the membrane proteins. Now, so what is the trick? You basically take now advantage of one particular property of these protein-containing nanodiscs that the other free nanodiscs without protein don't have. It could be, for instance, those that contain your protein. The protein would be very charged. And you take basically a matrix that has the opposite charge. Then it would stick here. And the rest of those particles that don't carry the protein, you would wash away. That means now all your particles are in the column. They are sticked. But now you can make a trick. You can reduce this interaction again and free them. Do you have a suggestion? So let's say if we would have, for instance, electrostatic interaction. Tanmai would be negative. He is basically a little chemical group on the column that would be negatively charged, and I would be positively charged. So we would, due to electrostatic, interact. How can we reduce this interaction?
By reducing my charge.
By using?
By reducing my charge, making it more positive.
Yeah, then I would need to change you. That's not easy, because the column is filled with a material of, for instance, negatively charged side groups. And Tanmay suggested, Thomas, let's change this. In principle, a nice idea, but not easy to do, technically, not easy to do. There was this suggestion, the pH. So why would be so first of all, what means pH? It means the amount of protons in the solutions. Okay, so why would this eventually change the interaction between me and Tanmay based on electrostatics? It's screening, no? Exactly. And then basically the protons will replace me. So, because also when I'm bound, if I'm very strongly bound, then we have a problem. Then it's very difficult. But if I'm bound in a way that I can bind, but also leave, then I would bind, because we have, of course, many Tanmays in this column, a lot of positions where I can bind again. So there will be always an equilibrium that I bind and I'm off, bind and I'm off. The moment I'm off and there's a lot of protons, they will shield. And I will never go back. So we can play, for instance, with the pH. We can also play with the salt concentration. Yeah, if you overdo it, the protein structure will change, because also the protein structure is based, for instance, of electrostatic interactions between amino acids in the basically protein, and that will also then, of course, change eventually the conformation or the stability of the protein. So you have to find a window where basically you break this interaction, but do not yet interfere with the interactions in your protein. Otherwise, you would unfold it, and for biology, that means it's a dead protein. You can compare this with an egg. So if you take an egg, you have the yellow egg and the white egg, but when you heat it, then you basically have denaturated the egg, and you never get it back in the natural state. So this egg is dead. You cannot use it for anything anymore. Okay, the question was, when we want to purify here these nanodiscs that contain the proteins away from those that don't contain the protein, what technique could we use? And we discussed that could be affinity-based. So we need to think what is so special on the protein-containing ones compared to the others. It's the protein that is in, and now the protein has a particular properties. It could have, for instance, charges. It could be overall negatively charged, or we make it the other way, overall positively charged. Then it would have the tendency to bind to a negatively charged material. But this interaction is now, I'm sitting here, and you can purify everything away what this does not contain a protein. But how do we then get our protein-containing nanodiscs out of this column? And the trick is to weaken this interaction again. And one way would be to change the pH. Another way would be to use salt, to add more salt. What would happen if you add more sodium chloride to your buffer? I'm now bound, and you flush buffer around, so water with ions, sodium chloride, for instance. The sodium chloride is then dissociated into sodium and chloride ions. That would be basically here a negatively charged matrix. If I get off, then all the sodiums would bind here. And the more sodium is there, the more the chances that sodium would stay instead that I would bind. So that's a type of, we call it affinity. You reduce the affinity of your particle of interest to the matrix that this particle has bound. And by that you can purify, take advantage of basically these different properties to purify your particle. Okay, another technique would be these free flow electrophoreses. What we do there, we take advantage of the charges. So if you, for instance, take these particles and you add charged lipids to them, what would happen? What was these charged lipids that you can use from nature? PS. What would be the charge of phosphatidylserine? Negative. Remember, in biology, there's only negatively charged lipid existing or neutral charged lipids. Only when you go to synthetic lipids, you can make positively charged. Okay? So if you make these nanodiscs with a part of negatively charged lipids, they would be negatively charged. If this would be a chamber, where you have here an electrode, positively charged, and here an electrode, negatively charged. And you put voltage on. What would happen? Now you are, as physicists, there would be an electric field. What is the rule of physics? What happens to particles that have a charge in an electric field? Exactly. There is a force, and they will basically take. So, if I'm now the charged particle, and in the moment there's no voltage, meaning no electric field, I would just move in the chamber with the flow. It's a flow, it's a constant flow that drives me in the end to this side. And I will come out on that side. If I take a voltage in between, Tanma, you want to switch on the voltage then, just symbolically? So in the moment there's no voltage, Tanma is observing me, and now we switch on the voltage. What will happen to me? Okay, I would move here, but since I have also a flow like that, it will drag me on that, but it will also drag me on that side, so I will start to move here, right? And now you switch it off, and then I would stay here, right? If you, of course, keep the electric field on, all the particles will have moved here. So it's depending on the flow speed versus the time of the electric field, how fast I do move, whether I will basically, depending on my charge, then move to this position or this. Because what would happen if we do the same experiment? Now in the first experiment, I carried one negatively charged. Now I carry in this particle two negatively charged lipids. What would happen then in the electric field as soon as Tanmay now makes this on? Now I have two charges. What would it mean in the electric field? It would move. So depending on which charge this particle is carrying, I will end up in different positions at the end of the chamber. So you can basically collect these different samples. So you would basically have a collection point on that side, on this side, on that side, on that side, and you can collect at each position, and depending on the charge of this particle, on the strength of this electric field, you can basically get a type of separation of the particles. And that works very well, actually. Yes? Yes?
What is the strength of the electric field that we apply?
Yeah, I would need to calculate it. I can tell you it's 200 voltage. Now you can calculate, because what would you, oh, you are a physicist, I like this. So you have 200 voltage. Would this be enough to calculate the strength of the electric field? What would you need to know from me? What would be all parameters now important to basically calculate the electric field in this chamber? I like this question because biologists don't think about it, but sometimes it's good to think about it. Yes. And also the chamber, no? It's the geometry of the chamber. It makes a difference whether you have this and the electrodes or whether it's, no? So all of this you would need to know, and of course the people that have produced these chambers, they have exactly these parameters. So you can make very clear physics. You take all the electric equations that you know, and you put these parameters in. So I have not calculated what would be the strength of the electric field, but I can tell you the voltage. I could tell you the geometry of the chamber, and then you as a physicist could just sit down and calculate the strength of the electric field. So like voltage per square centimeter, whatever you would like to calculate. Yeah. Yes.
These nanodiscs will be in some buffer, right? They will have salt. And due to that, there will be electrical double layer formation and clay.
Yeah, what a disaster, no?
Like there is a field gradient.
Absolutely.
So due to field gradient, it experiences different force, and hence it is experiencing different force at each point, and hence there is a deformation of nanodisc, right?
Yeah, if it could happen, and you are fully right. Since you do it in a buffer of salt, it's not easy, because eventually you would like to have, of course, only a solution that is unconductive. Because now if you start to have a conductive fluid, what would be the problem? When ions start also to move in this electric field, you get heat. You heat up actually the solution. So you need to cool this system, because otherwise you destroy. Because you are right, we cannot work without ions. Because our proteins, they like also ions for stability, to be stabilized. So we need to work in a buffer that on top of water contains ions and our particle. And you will create basically also a flow of ions in the chamber, and you will create basically not such a nice, there's a gradient based on this, also on your ions that are moving. In reality, we almost don't care, because we are only interested that we separate, so we need to find the right conditions of the voltage, how much would be the strength that we want. We need to be careful with the buffer. We might need to use a low amount of ion concentration there, which could be a danger for your protein again. So we don't work under physiological buffer conditions. We don't use this ocean water. We don't use 150 millimolar sodium chloride in these solutions. That would be too strong. You are fully right. Because it's not an isolator that it's not a condenser that we have created there, it's a conductive fluid that we have. Very good. I mean, all these points are, if you want to work with this, it's good to have this type of understanding, because that allows you also to play a little bit on the parameter, and on the same time you would need to talk with biologists. You know, Thomas, I want to have this amount only on ions, because I calculated if I have more ions, the heating would be too much. And then I would say, okay, Tanmay, yes, but don't go below 50 millimolar, because we have found that then our protein gets inactivated. Yeah? And then you can still, yeah, that could be still good enough for this type of experiment we can do. Or you say, Thomas, this is still not a good condition. Then we cannot use this technique. Yeah? That's important. But you see also how important it is that you have an understanding of physics, a little bit of biology. You need to have understanding of chemistry, because that helps you to talk to the people. Yeah? I see also often people talking. But since you basically just have an idea of what would be, from the point of biological, an interesting question and what could be a problem there, that helps you in designing basically, for instance, your type of experiments in terms of modeling or in terms of technical development. I mean, this is, for instance, also something people are pretty much interested as a technical development. No? And you see already, I mean, people were also thinking to use this in the medical basis, but it's not so easy, because in the medical basis, you want to have fast results and you want to have an easy system. So the best is basically, you know, you have a type of paper stripe, and then ill or not ill. No? But that would be that's not always the case. But from the technical point, if you can develop something like that, that will be the best seller, because everyone can use it, cancer or not. So in the morning, and then you get already your indications. Oh, today you are, you know, better careful. Something is wrong with your body, or you should better go to the doctor. Even if the stripe does not tell you what you have, but at least it can tell you, please go to the doctor to check. And the doctor can take a blood sample to get more information. What could be the reason that, you know, some indicator has basically reacted. Okay. Okay. Good. So we could basically use affinity, we could use this technique, and now you can imagine, let's say we take such a nanodisc, and we prepare them first without protein, and we would have maybe, you know, half uncharged, half charged lipids. If I now basically prepare the same nanodisc with a protein in, what will be the consequence? There will be less lipids in. What would this mean for the charge? It will be less charged, this particle. Or the opposite, your protein is supercharged, and you put in an uncharged nanodisc a charged protein, then the particle becomes charged. So that means you can also use this technique not only to separate nanodiscs of different lipid composition, but also nanodiscs, whether they contain a protein or not. Because the protein will also influence the overall charge, and that depends only whether the protein is charged and how many charged lipids are left when you put the protein in. Because when you have the protein in, less lipids, there's less space for the lipids left. Okay, so this is just shown here. So here we make basically different nanodiscs. Nanodiscs that are not charged and nanodiscs that are very charged. And then we collected here in the end always these fractions. Yeah? And then you see, depending on the travel path, you can separate them. So if they are uncharged, they hardly travel to the side. So they stay more like that. If they are a bit charged, they will in a short time, when you have this on-off period of the electric field, move a little bit. The others that are most charged, no? So you collect and you can basically separate them. Yeah? Maybe you are not impressed by the separation, because you would hope that this would be, everything would be basically on that side and everything would be on that side. For biologists, this is already very good, I tell you. So don't be disappointed. It's not so easy, because the changes are also are small in terms of electrocharge differences between, for instance, a nanodisc with a protein and without a protein. And the reason why these changes are small, we talked, if you have a buffer with ions, ions start to shield also, right? So it's not, you can only make it extreme if you want to say very negatively charged, this is phosphatidylglycerol, negatively charged, so very, very negatively charged not, then it's quite, there's quite a difference. But if these changes are more smaller in the terms of lipid composition, then also the separation is less good. Okay, and here you see an example for nanodiscs with basically a protein and without a protein. They have the same lipids, only they differ with protein, without. And you see there's basically, here's the portion of the nanodiscs that do not contain, and you have a portion that contains. You know, even those that contain a protein, some of those end up. But you see that, let's say, this shoulder here, there's hardly any of those that do not contain anymore. So if you only collect those, then you have a very poor sample. With the disadvantage, you have lost a lot of material. And that's also something for biologists. You know, we start with a lot of material, and then we lose. After we purify, and we lose. Maybe for those, I don't know whether you needed to do here some chemistry. When I was a chemistry student, we needed also to do organic chemistry. So some simple reactions. And then the chemist has something very important. They always say, how much did you get out? And I know that at that time, it was almost like an exam. In chemistry, we called it cooking. So we needed to cook. And then we needed to determine how much did we get out. And then it was basically written, you have to get out 1 gram. And then the teacher was very sharp. Thomas, you don't have synthesized 1 gram. Go back to the lab. You have done something wrong. There was something not optimal in your reactions. The yield is not high enough. So for chemists, it's always the yield. You want to have, of course, a higher yield. You want to have those components here to react, and you get 100% out. You put basically 2 substances in, and all of this is converted into your dream substance that you wanted to produce. In reality, that doesn't happen, and sometimes it's even very low. That means you need to invest a lot of money for the substrate, and you end up with a little. And that you can also sometimes see on the prices, something which is very difficult to synthesize. Many steps, you lose and lose. You wonder, you know, 1 milligram, why is it so costly? Because you have used so much material before to get it. Yeah? So for chemists, it's also the yield very important, like for biochemists. Yeah, Tripta is not here, I can tell. Tripta, Thomas sent us vesicles with a lot of protein. Yeah? And I say, oh, it's not so easy. You know, we need for the experiments, because also for the experiments, you need some material. And that's something basically where you might not appreciate. You need to produce the protein, but then you typically need to purify it. It's contaminated. Or here, you need to purify it in these nanodiscs that contain it. Okay, good. So now basically we talked about this approach. We have basically our bait proteins, and we can basically mix them with our protein of interest. But why not do something else? Why not take maybe a biological membrane? We could take also a biological membrane. We don't need to start from the purified protein. So we could take a biological membrane, then solubilize, add detergent, and we have our proteins. Those proteins we could add all in nanodiscs. Or we purify the protein of interest that we are interested in, and then we basically reconstitute, but now not only with lipids, but plus the bad proteins. Okay? So in a way you can do 2 things. You can use the biological membrane, solubilize it, and have a protein mix. And there was one thing, if you now to this protein mix would add lipids back and the bad protein, what do you think will happen when you do this? Let's say you are the biological membrane. Now you are protein, lipids. And I add detergent just for what would happen? The detergent molecules would start to enter here, would destroy this interaction of the lipids, would start to destroy the interaction of the protein with the lipids, so you would be all free. Now I add more lipids, belt proteins, and I remove the detergents. What would just, if you imagine, what would happen? What could happen? Okay, and how reassemble? How would it reassemble? Tanma, you are still okay? No, you stay, but you are still that I make some experiments with you? Okay, so no, we would have many of those belt proteins swimming. So this belt protein would reassemble with lipids and a protein. No? Then we can basically, here's this other copy of the belt protein. No? What could it do? It could go here. Right? Could it also do this?
Yes, sir. If it can accommodate, then it can do.
Here was a very, he said, yes, but if it can, if it's large enough. Otherwise not. Because, you see already I have a problem here. I don't get all three of you. That will not work. But can we not take advantage of this? Sorry, Tanmay. Can we not take an advantage of this? We would basically put each individual protein into a nanodisc. You would be in one disc. You would be in another disc. You would be in another disc. You have one charge. You have another charge. You are overall not charged. We can start to separate you out of a mixture of proteins just that we have taken from the membranes, right? So you could basically take this also as a type of purification without a pre-purification. Yeah? Okay, so that is possible, but this is still maybe something useful, also used, but there's still one disadvantage. Do you remember? We add detergent. What was this problem with adding detergent? It can interfere. It disrupts, no? It disrupts these lipid-protein interactions, maybe protein-protein interactions. It's harsh. Could we not think about something? There was also, I think someone asked me in the course, Thomas, can we not directly take the membrane and take the protein out with all of these detergents? We almost like, do you like to eat cake? Yeah. So when you get guests and you have a big cake, what do? You cut the cake into pieces with a cookie cutter, a knife, right? So would it not be fantastic if I would have something like this knife and you are sitting here in this membrane, you as a protein, you, and I cut this guy out and I take him out without detergent. I have not added detergent. I have just taken, and what would I take eventually with him? Some lipids. I would cut him out with lipids. Would this not be fantastic? And this is what people, of course, wanted. Right? And they came up with a solution. So there are particular polymers, copolymers, small molecules that have this ability to actually get into the membrane, and if you add enough, even to take pieces out of the membrane. So what these properties, what would you expect if you would need to talk to a chemist? What do you think the chemist will say? Okay, what type of polymer would you need to synthesize? Just from the properties, should it be a water-soluble polymer? Water-soluble? Lipid-soluble? Only lipid-soluble? Should be something in between again, no? Something like amphiphile. It should like lipids, but also should like water. No? So these copolymers are again like this amphiphilic character. They have basically a size that likes water, and they have a size that likes lipids, and with the size that likes lipids, they would start to wrap around and eventually take out a piece. Yeah? And then you see, basically, here was the detergent. Here's no detergent. No detergent anymore needed. So you cut out the membrane from the environment where, membrane protein from the environment where it sits with the lipids around. It's also an interesting technique because if these lipids are very tightly bound, you see here's a lipid with this membrane protein, I cut out basically and I get a feeling also what type of lipids are with you. Because as we discussed, it could be that these proteins just unspecifically interact, sometimes meeting one type of lipid, sometimes meeting another type of lipid. In reality, often these membrane proteins have a preference for particular proteins. We call it the lipid annulus. So lipids that stay with this protein. A little bit like ions in water that have a hydration shell. They carry water with them, right? And also often membrane proteins that are sitting are not basically just randomly interacting, but some lipids preferentially interact. What could be reasons for that? If you remember a little bit maybe the aspects that we discussed, that membrane proteins would like to have particular lipids around them. It could be, for instance, the size of the hydrophobic part of the protein. When this protein would be in a lipid mixture, it would be preferentially interact with lipids with a long hydrophobic part. No? So that would basically shield the hydrophobic part. And then you get a type of demixing. Some lipids would be preferentially associated with your protein. Okay, good. So now detergent, you might even get, if the copolymers are long enough, protein complexes out of the membrane. And you get type of native-like membranes with the protein isolated. And here are just few examples. You don't need really to basically learn them, but it's basically a polymer means you have basically chemical components repeatedly repeat. And that would be here a hydrophobic part, and that would be a hydrophilic part, you see? So this likes the lipids, this likes the water phase, and that's why they would start to wrap around your basically membrane particles, or even have this ability first to cut out a piece of membrane when you add those in an access to your biological membrane of interest. Yeah, what can you then do? So you can take our classical nanodiscs, or you take these nanodiscs with a copolymer, and you can, for instance, now when you have isolated in these nanodiscs your protein, you can immobilize them on a type of surface where you can sensitive measure whether what in terms of how many particles have been actually attached, and whether additional particles, because these could be proteins that interact with your membrane protein or that interact with your membrane, they would attach and they would induce a change. Or you can do, for instance, scattering techniques. So you can basically use type of electron scattering, neutron scattering, and you can get informations because you can get an assumption how the particles should look like or could look like. You model this and you get a scattering pattern. Then you compare this scattering pattern with the scattering pattern that you have obtained, and that will give you an idea whether, for instance, your protein is in a complex like that, or maybe is in a complex more like that, because the structure will be different of this particle compared to this particle. And this allows you also very nice protein-protein interaction structural studies. Yeah, with this I would like to end, and I would give you then tomorrow some, or maybe two examples how you can use this in a more detail to study